Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Flagellum. 56 languages. Flagellum. Structure of bacterial flagellum. SEM image of flagellated eukaryote. Chlamydomonas sp. 10000. Identifiers. Mesh D005407. THH1.00.01.1.01032. FMA 67472. Anatomical terminology. For the insect anatomical structure, see antenna, biology. For the flagella of male solifugae, see solifugae. For eukaryotic only, see cilia. A flagellum, /FLDLM/PL, flagella, Latin for whip or scourge, is a hair-like appendage that protrudes from certain plant and animal sperm cells, from fungal spores, zoospores, and from a wide range of microorganisms to provide motility. 1, 2, 3, 4. Many protists with flagella are known as flagellates. A microorganism may have from one to many flagella. A gram-negative bacterium Helicobacter pylori, for example, uses its flagella to propel itself through the stomach to reach the mucous lining where it may colonize the epithelium and potentially cause gastritis and ulcers, a risk factor for stomach cancer. 5. In some swarming bacteria, the flagellum can also function as a sensory organelle, being sensitive to wetness outside the cell. 6. Across the three domains of bacteria, archaea, and eukaryota, the flagellum has a different structure, protein composition, and mechanism of propulsion, but shares the same function of providing motility. The Latin word flagellum means whip to describe its lash-like swimming motion. The flagellum in archaea is called the arculum to note its difference from the bacterial flagellum. 7, 8. Eukaryotic flagella and cilia are identical in structure, but have different lengths and functions. 9. Prokaryotic fimbriae and pili are smaller and thinner. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Edit on Wikidata. Prokaryotic, bacterial and archaeal flagella run in a rotary movement, while eukaryotic flagella run in a bending movement. The prokaryotic flagellum uses a rotary motor, and the eukaryotic flagellum uses a complex sliding filament system. Eukaryotic flagella are ATP driven, while prokaryotic flagella can be ATP driven, archaea, or proton driven. Bacteria. 11. Appendages, with different functions. Cilia are attached to the surface of flagella and are used to swim or move fluid from one region to another. 10. Types, edit. The three types of flagella are bacterial, archaeal, and eukaryotic. The flagella in eukaryotes have dynein and microtubules that move with a bending mechanism. Bacteria and archaea do not have dynein or microtubules in their flagella, and they move using a rotary mechanism. 12. Other differences among these three types are: bacterial flagella are helical filaments, each with a rotary motor at its base which can turn clockwise or counterclockwise. 13, 14, 15, they provide two of several kinds of bacterial motility. 16, 17. Archaeal flagella, arcula, are superficially similar to bacterial flagella in that it also has a rotary motor, but are different in many details and considered non-homologous. 18, 19, 20. Eukaryotic flagella, those of animal, plant, and protist cells, are complex cellular projections that lash back and forth. Eukaryotic flagella and motile cilia are identical in structure, but have different lengths, waveforms, and functions. Primary cilia are immotile, and have a structurally different 9+0 axon rather than the 9+2 axon found in both flagella and motile cilia. Bacterial flagella, edit. Structure and composition, edit. The bacterial flagellum is made up of protein subunits of flagellin. 12. Its shape is a 20 nanometer thick hollow tube. It is helical and has a sharp bend just outside the outer membrane. This hook allows the axis of the helix to point directly away from the cell. A shaft runs between the hook and the basal body, passing through protein rings in the cell's membrane that act as bearings. Gram-positive organisms have two of these basal body rings, one in the peptidoglycan layer and one in the plasma membrane. Gram-negative organisms have four such rings. The L ring associates with the lipopolysaccharides, the P ring associates with peptidoglycan layer, the M ring is embedded in the plasma membrane, and the S ring is directly attached to the cytoplasm. The filament ends with a capping protein. 21, 22. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Bacterial flagella motor assembly, shown here is the C ring at the base with Flig in red, Fl-M in yellow, and Fl-N in shades of purple, the MS ring in blue, the MoAB in brown, the LP ring in pink, and the rod in gray. 25. The flagellar filament is the long, helical screw that propels the bacterium when rotated by the motor, through the hook. In most bacteria that have been studied, including the gram-negative Escherichia coli, Salmonella typhimurium, Caulobacter crescentus, and Vibrio algenolyticus, the filament is made up of 11 protofilaments approximately parallel to the filament axis. Each protofilament is a series of tandem protein chains. However, Campylobacter jejuni has 7 protofilaments. 23. The basal body has several trays in common with some types of secretory pores, such as the hollow, rod-like plug in their centers extending out through the plasma membrane. The similarities between bacterial flagella and bacterial secretory system structures and proteins provide scientific evidence supporting the theory that bacterial flagella evolved from the type 3 secretion system. TTSS. The atomic structure of both bacterial flagella as well as the TTSS injectism have been elucidated in great detail, especially with the development of cryo-electron microscopy. The best understood parts are the parts between the inner and outer membrane, that is, the scaffolding rings of the inner membrane, IM, the scaffolding pairs of the outer membrane, OM, and the rod/needle. Injectism, or rod/hook, flagellum, sections. 24. Motor, edit. Further information, rotating locomotion in living systems. The bacterial flagellum is driven by a rotary engine, MO. complex, made up of protein. Located at the flagellum's anchor point on the inner cell membrane. The engine is powered by proton motive force, i.e., by the flow of protons, hydrogen ions, across the bacterial cell membrane due to a concentration gradient set up by the cell's metabolism. Vibrio species have two kinds of flagella, lateral and polar, and some are driven by a sodium ion pump rather than a proton pump. 26. The rotor transports protons across the membrane, and is turned in the process. The rotor alone can operate at 6,000 to 100,000 rpm. 27, but with the flagella filament attached usually only reaches 200 to 1000 rpm. The direction of rotation can be changed by the flagellar motor switch almost instantaneously, caused by a slight change in the position of a. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Protein, Flig, in the rotor. 28. The torque is transferred from the MoAB to the torque helix on Flig's D5 domain, and with the increase in the requirement of the torque or speed, more MoAB are employed. 25. Because the flagellar motor has no on-off switch, the protein Epsi is used as a mechanical clutch to disengage the motor from the rotor, thus stopping the flagellum and allowing the bacterium to remain in one place. 29. The production and rotation of a flagellum can take up to 10% of an Escherichia coli cell's energy budget and has been described as an energy-guzzling machine. 30. Its operation generates reactive oxygen species that elevates mutation rates. 30. The cylindrical shape of flagella is suited to locomotion of microscopic organisms. These organisms operate at a low Reynolds number, where the viscosity of the surrounding water is much more important than its mass or inertia. 31. The rotational speed of flagella varies in response to the intensity of the proton motive force, thereby permitting certain forms of speed control, and also permitting some types of bacteria to attain remarkable speeds in proportion to their size. Some achieve roughly 60 cell lengths per second. At such a speed, a bacterium would take about 245 days to cover 1 kilometer. Although that may seem slow, the perspective changes when the concept of scale is introduced. In comparison to macroscopic life forms, it is very fast indeed when expressed in terms of number of body lengths per second. A cheetah, for example, only achieves about 25 body lengths per second. 32. Through use of their flagella, bacteria are able to move rapidly towards attractants and away from repellents, by means of a biased random walk, with runs and tumbles brought about by rotating its flagellum counterclockwise and clockwise, respectively. The two directions of rotation are not identical, with respect to flagellum movement, and are selected by a molecular switch. 33. Clockwise rotation is called the traction mode with the body following the flagella. Counterclockwise rotation is called the thruster mode with the flagella lagging behind the body. 34. Assembly, edit. During flagellar assembly, components of the flagellum pass through the hollow cores of the basal body and the nascent filament. During assembly, protein components are added at the flagellar tip, rather than at the base. 35. In vitro, flagellar filaments assemble spontaneously in a solution containing purified flagellin as the sole protein. 36. Evolution, edit. Main article, Evolution of flagella. At least 10 protein components of the bacterial flagellum share homologous proteins with the type 3 secretion system, T3SS, found in many gram-negative bacteria, 37, hence one likely evolved from the other. Because the T3SS has a similar number of components as a flagellar apparatus. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. About 25 proteins, which one evolved first is difficult to determine. However, the flagellar system appears to involve more proteins overall, including various regulators and chaperones, hence it has been argued that flagella evolved from a T3SS. However, it has also been suggested, 38, that the flagellum may have evolved first or the two structures evolved in parallel. Early single-cell organisms need for mobility, support that the more mobile flagella would be selected by evolution first, 38. But the T3SS evolving from the flagellum can be seen as reductive evolution, and receives no topological support from the phylogenetic trees. 39. The hypothesis that the two structures evolved separately from a common ancestor accounts for the protein similarities between the two structures, as well as their functional diversity. 40. Flagella and the Intelligent Design Debate, edit. Main articles, Intelligent Design and Irreducible Complexity. Some authors have argued that flagella cannot have evolved, assuming that they can only function properly when all proteins are in place. In other words, the flagellar apparatus is irreducibly complex. 41. However, many proteins can be deleted or mutated and the flagellum still works, though sometimes at reduced efficiency. 42. Moreover, with many proteins unique to some number across species, diversity of bacterial flagellar composition was higher than expected. 43. Hence, the flagellar apparatus is clearly very flexible in evolutionary terms and perfectly able to lose or gain protein components. For instance, a number of mutations have been found that increase the motility of E. coli. 44. Additional evidence for the evolution of bacterial flagella includes the existence of vestigial flagella, intermediate forms of flagella and patterns of similarities among flagellar protein sequences, including the observation that almost all of the core flagellar proteins have known homologies with non-flagellar proteins. 37. Furthermore, several processes have been identified as playing important roles in flagellar evolution, including self-assembly of simple repeating subunits, gene duplication with subsequent divergence, recruitment of elements from other systems, molecular bricolage, and recombination. 45. Flagellar arrangements, edit. Different species of bacteria have different numbers and arrangements of flagella, 46, 47, named using the term tricho, from the Greek trichos meaning hair. 48. Monotrichous bacteria such as Vibrio cholerae have a single polar flagellum. 49. Amphitrichous bacteria have a single flagellum on each of two opposite ends, for example, Campylobacter jejuni or Alcaligens faecalis, both flagella rotate but coordinate to produce coherent thrust. Lophorichous bacteria, lopho Greek combining term meaning crest or tuft, 50, have multiple flagella located at the same spot on the bacterial surface such as Helicobacter pylori, which act in concert to drive the bacteria in a single direction. In many cases, the bases of multiple flagella. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Are surrounded by a specialized region of the cell membrane, called the polar organelle. Citation needed. Paritrichus bacteria have flagella projecting in all directions, for example, E. coli. Counterclockwise rotation of a monotrichus polar flagellum pushes the cell forward with the flagellum trailing behind, much like a corkscrew moving inside cork. Water on the microscopic scale is highly viscous, unlike usual water. Spirochetes, in contrast, have flagella called endoflagella arising from opposite poles of the cell, and are located within the periplasmic space as shown by breaking the outer membrane and also by electron cryotomography microscopy. 51. The rotation of the filaments relative to the cell body causes the entire bacterium to move forward in a corkscrew-like motion, even through material viscous enough to prevent the passage of normally flagellated bacteria. In certain large forms of Selenomonas, more than 30 individual flagella are organized outside the cell body, helically twining about each other to form a thick structure, easily visible with the light microscope, called a fascicle. In some Vibrio spp, particularly Vibrio parahemolyticus, 52, and related bacteria such as Aeromonas, two flagellar systems coexist, using different sets of genes and different ion gradients. For energy, the polar flagella are constitutively expressed and provide motility in bulk fluid, while the lateral flagella are expressed when the polar flagella meet too much resistance to turn. 53, 54, 55, 56, 57, 58. These provide swarming motility on surfaces or in viscous fluids. Bundling, edit. Bundling is an event that can happen in multiflagellated cells, bundling the flagella together and causing them to rotate in a coordinated manner. Flagella are left-handed helices, and when rotated counterclockwise by their rotors, they can bundle and rotate together. When the rotors reverse direction, thus rotating clockwise, the flagellum unwinds from the bundle. This may cause the cell to stop its forward motion and instead start twitching in place, referred to as tumbling. Tumbling results in a stochastic reorientation of the cell, causing it to change the direction of its forward swimming. It is not known which stimuli drive the switch between bundling and tumbling, but the motor is highly adaptive to different signals. In the model describing chemotaxis, movement on purpose, the clockwise rotation of a flagellum is suppressed by chemical compounds favorable to the cell, for example, food. When moving in a favorable direction, the concentration of such chemical attractants increases and therefore tumbles are continually suppressed, allowing forward motion. Likewise, when the cell's direction of motion is unfavorable, for example, away from a chemical attractant, tumbles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Eukaryotic flagella. 1. Axoneme, 2 cell membrane, 3. IFT, intraflagellar transport, 4. Basal body, 5 cross-section of flagella, 6 triplets of microtubules of basal body. Cross-section of an axoneme. Longitudinal section through the flagellar area in Chlamydomonas reinhardtii. In the cell apex is the basal body, that is, The anchoring site for a flagellum. Basal bodies originate from and have a substructure similar to that of centrioles, with 9 peripheral microtubule triplets. See structure at bottom center of image. The 9+2 structure is visible in this cross-section. Micrograph of an axoneme are no longer suppressed and occur much more often, with the chance that the cell will be thus reoriented in the correct direction. Even if all flagella would rotate clockwise, however, they often cannot form a bundle due to geometrical and hydrodynamic reasons. 59, 60. Eukaryotic flagella, edit. Terminology, edit. Aiming to emphasize the distinction between the bacterial flagella and the eukaryotic cilia and flagella. Some authors attempted to replace the name of these two eukaryotic structures with undulipodia, for example, all papers by Margulies since the 1970s, 61, or cilia for both. For example, Hulsman, 1992, 62, ADL et al., 2012, 63, most papers of Cavalier-Smith, preserving flagella for the bacterial structure. However, the discriminative usage of the terms cilia and flagella for eukaryotes adopted in this article, see flagella versus cilia below, is still common. For example, Anderson et al. 1991, 64, Leadbeater et al. 2000. 65. Internal structure, edit. The core of a eukaryotic flagellum, known as the axoneme, is a bundle of 9 fused pairs of microtubules, known as doublets, surrounding 2 central single microtubules, singlets. This 9+2 axoneme is characteristic of the eukaryotic flagellum. At the base of a eukaryotic flagellum is a basal body, blepharoplast, or kinetosome, which is the microtubule organizing center for flagella microtubules and is about 500 nanometers long. Basal. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Beating pattern of eukaryotic flagellum and xylem, a traditional distinction before the structures of the two are known. Bodies are structurally identical to centrioles. The flagellum is encased within the cell's plasma membrane, so that the interior of the flagellum is accessible to the cell's cytoplasm. Besides the axoneme and basal body, relatively constant in morphology, other internal structures of the flagellar apparatus are the transition zone, where the axoneme and basal body meet, and the root system, microtubular or fibrillar structures that extend from the basal bodies into the cytoplasm, more variable and useful as indicators of phylogenetic relationships of eukaryotes. Other structures, more uncommon, are the paraflagellar, or paraxial, paraxonemal, rod, the R fiber, and the S fiber. 66, 63, 84 for surface structures, see below. Mechanism, edit. Each of the outer 9 doublet microtubules extends a pair of dynein arms, an inner and an outer arm, to the adjacent microtubule. These produce force through ATP hydrolysis. The flagellar axoneme also contains radial spokes, polypeptide complexes extending from each of the outer 9 microtubule doublets towards the central pair, with the head of the spoke facing inwards. The radial spoke is thought to be involved in the regulation of flagellar motion, although its exact Function and method of action are not yet understood. 67. Flagella versus cilia, edit. The regular beat patterns of eukaryotic cilia and flagella generate motion on a cellular level. Examples range from the propulsion of single cells such as the swimming of spermatozoa to the transport of fluid along a stationary layer of cells such as in the respiratory tract. 68. Although eukaryotic cilia and flagella are ultimately the same, they are sometimes classed by their pattern of movement, a tradition from before their structures have been known. In the case of flagella, the motion is often planar and wave-like, whereas the motile cilia often perform a more complicated three-dimensional motion with a power and recovery stroke. 68. Yet another traditional form of distinction is by the number of 9+2 organelles on the cell. 67. Intraflagellar transport, edit. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Intraflagellar transport, the process by which axonal subunits, transmembrane receptors, and other proteins are moved up and down the length of the flagellum, is essential for proper functioning of the flagellum, in both motility and signal transduction. 69. Evolution and occurrence, edit. Further information, evolution of flagella. Eukaryotic flagella or cilia, probably an ancestral characteristic, 70, are widespread in almost all groups of eukaryotes, as a relatively perennial condition, or as a flagellated life cycle stage, for example, zooids, gametes, zoospores, which may be produced continually or not. 71, 72, 63. The first situation is found either in specialized cells of multicellular organisms, for example, the choanocytes of sponges, or the ciliated epithelia of metazoans, as in ciliates and many eukaryotes. With a flagellate condition, or monadoid level of organization, see flagellator, an artificial group. Flagellated life cycle stages are found in many groups, for example, many green algae, zoospores and male gametes, bryophytes, male gametes, pteridophytes, male gametes, some gymnosperms, cycads, and ginkgo, as male gametes, centric diatoms, male gametes, brown algae, zoospores and gametes, eumystes, asexual zoospores and gametes, hyphochytrids, zoospores, labyrinthulomystes, zoospores, some apicomplexans, gametes, some radiolarians, probably gametes. 73, foraminifrons, gametes, plasmodiaphromystes, zoospores and gametes. Myxogastrids, zoospores, metazoans, male gametes, and chytrid fungi, zoospores and gametes. Flagella or cilia are completely absent in some groups, probably due to a loss rather than being a primitive condition. The loss of cilia occurred in red algae, some green algae, Zineumatophyceae. The gymnosperms except cycads and ginkgo, angiosperms, pennate diatoms, some apicomplexans, some amoebozoans, in the sperm of some metazoans, 74, and in fungi, except chytrids. Typology, edit. A number of terms related to flagella or cilia are used to characterize eukaryotes. 72, 75, 66, 60, 63, 76, 77. According to surface structures present, flagella may be whiplash flagella, equals smooth, achromatic flagella without hairs, for example, in Opisthoconta. Hairy flagella equals tinsel, flimmer. Pleuronematic flagella with hairs equals Mastigonum sensuolato. Divided in. With fine hairs equals non-tubular or simple hairs occurs in Eugalnophaceae, Dinoflagellata. Some Haptophaceae, Pavlovals. With stiff hairs equals tubular hairs, Retromes, Mastigonum sensu stricto, divided in. Bipartite hairs, with two regions. Occurs in Cryptophaceae, Prasonophaceae, and some Heteroconta. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Tripartite equals Straminopilis, hairs, with three regions, a base, a tubular shaft, and one or more terminal hairs. Occurs in most Heteroconta. Stechonomatic flagella, with a single row of hairs. Pantomatic flagella, with two rows of hairs. Achromatic flagella with a single, terminal mastigonum or flagellar hair, for example, Bodonids, 78. Some authors use the term as synonym of whiplash. With scales, for example, Prasonophaceae. With spines, for example, some brown algae. With undulating membrane, for example, some kinetoplastids, some parabasalids. With proboscis, trunk-like protrusion of the cell, for example, Apusimonids, some Bodonids, 79. According to the number of flagella, cells may be, remembering that some authors use ciliated instead of flagellated, 63, 80. Uniflagellated, for example, most Opisthoconta. Biflagellated, for example, all Dinoflagellata, the gametes of Charophaceae, of most bryophytes and of some metazoans, 74. Triflagellated, for example, the gametes of some foraminifera. Quadriflagellated, for example, some Prasonophaceae, Collodictionidae. Octoflagellated, for example, some Diplomonida, some Prasonophaceae. Multiflagellated, for example, Opalinata, Ciliifera, Stephanopogon, Parabasalida, Hemimastigophora. Caryoblasti, multicilia, the gametes, or zoids, Ovidogonials, Chlorophyta, some Pteridophytes and some gymnosperms. According to the place of insertion of the flagella, 81. Opisthocont, cells with flagella inserted posteriorly, for example, in Opisthoconta, Vischer, 1945. In Haptophaceae, flagella are laterally to terminally inserted, but are directed posteriorly during rapid swimming. 82. Acrocont, cells with flagella inserted apically. Subacrocont, cells with flagella inserted subapically. Pleurocont, cells with flagella inserted laterally. According to the beating pattern. Gliding, a flagellum that trails on the substrate, 79. Heterodynamic, flagella with different beating patterns, usually with one flagellum functioning in food capture and the other functioning in gliding, anchorage, propulsion or steering, 83. Isodynamic, flagella beating with the same patterns. Other terms related to the flagella type. Isocont, cells with flagella of equal length. It was also formerly used to refer to the chlorophyta. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Anisocont, cells with flagella of unequal length, for example, some Eugalnophaceae and Prasonophaceae. Heterocont, term introduced by Luther, 1899, to refer to the Xanthophaceae, due to the pair of flagella of unequal length. It has taken on a specific meaning in referring to cells with an anterior straminopilis flagellum, with tripartite mastigonums, in 1 or 2 rows, and a posterior, usually smooth flagellum. It is also used to refer to the taxon Heteroconta. Stephanocont, cells with a crown of flagella near its anterior end, for example, the gametes and spores. Ovidogonials, the spores of some bryopsidales. Term introduced by Blackman and Tansley. 1902, to refer to the oedogonials. Acant, cells without flagella. It was also used to refer to taxonomic groups, as Aconta or Acanta. The Zeneumatophaceae and Bacillariophaceae, Altmans, 1904, or the Rhodophaceae. Christensen, 1962. Archaeal flagella, edit. The archaeolum possessed by some species of archaea is superficially similar to the bacterial flagellum. In the 1980s, they were thought to be homologous on the basis of gross morphology and behavior. 84. Both flagella and archaea consist of filaments extending outside the cell, and rotate to propel the cell. Archaeal flagella have a unique structure which lacks a central channel. Similar to bacterial type 4 pilins, the archaeal proteins, archaeolins, are made with class 3 signal peptids, and they are processed by a type 4 propylene peptidase-like enzyme. The archaeolins are typically modified by the addition of N-linked glycans which are necessary for proper assembly or function. 3. Discoveries in the 1990s revealed numerous detailed differences between the archaeal and bacterial flagella. These include: bacterial flagella rotation is powered by the proton motive force, a flow of H and ions, or occasionally by the sodium motive force, a flow of Na and ions. Archaeal flagella rotation is powered by ATP. 85. While bacterial cells often have many flagellar filaments, each of which rotates independently, the archaeal flagellum is composed of a bundle of many filaments that rotates as a single assembly. Bacterial flagella grow by the addition of flagellin subunits at the tip. Archaeal flagella grow by the addition of subunits to the base. Bacterial flagella are thicker than archaea, and the bacterial filament has a large enough hollow tube inside that the flagellin subunits can flow up the inside of the filament and get added at the tip. The archaeolum is too thin, 12 to 15 nm, to allow this. 86. Many components of bacterial flagella share sequence similarity to components of the type 3 secretion systems, but the components of bacterial flagella and archaea share no sequence similarity. Instead, some components of archaea share sequence and morphological similarity. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. With components of type 4 pili, which are assembled through the action of type 2 secretion systems, the nomenclature of pili and protein secretion systems is not consistent. 86. These differences support the theory that the bacterial flagella and archaea are a classic case of biological analogy, or convergent evolution, rather than homology. 87, 88, 89. Research into the structure of archaea made significant progress beginning in the early 2010s, with the first atomic resolution structure of an archaea protein, the discovery of additional functions of archaea, and the first reports of archaea in Nanorchaeota and Thaumarchaeota. 90, 91. Fungal, edit. The only fungi to have a single flagellum on their spores are the chytrids. In Batrachochytrium, Dendrobatid is the flagellum is 19-20 m long. 92. A non-functioning centriole lies adjacent to the kinetosome. 9 interconnected props attach the kinetosome to the plasmalemma, and a terminal plate is present in the transitional zone. An inner ring-like structure attached to the tubules of the flagellar doublets within the transitional zone has been observed in transverse section. 92. Additional images, edit. Multiple flagella in Lophorichus. Arrangement on surface of Helicobacter pylori. Physical model of a bacterial flagellum. See also, edit. Ciliopathy. RPOF. References, edit. 1 Bardi, Sonia L. Ing, Sundhi Y.M. Charl, Ken F. 1 February 2003. Prokaryotic motility. Structures. Microbiology. 149, 2295304 do. 10.1099 mic 0.25948-0. PMID 126249192. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 2 Silflo, Caroline D. Lefebvre, Paulo. 1 December 2001. Assembly and motility of eukaryotic cilia and flagella. Lessons from Chlamydomonas reinhardtii. Plant physiology. 127, for 1507. Do. 10.1104/pp.010807. PMC 1540183. PMID 1174394. 3 AB Jarl, Ken F., ed. 2009. Pili and flagella, Current Research and Future Trends. Norfolk, Caister. Academic Press. ISBN 978-1-904455-48-6. 4 Mallow, Aurelio F., Gomendio, Montserrat, God, Julian, Langlenton, Barbara, Sola, Anna J., Roldan. Eduardo R.S., the 22nd of June, 2006. Sperm Design and Sperm Function. Biology Letters. 2, 2246-249. Do. 10.1098/RSBL.2006.0449. PMC 1618917. PMID 1714837-4. 5 Lacey, B., Rosemore, J., October 2001. Helicobacter pylori, ulcers and more, the beginning of an era. The Journal of Nutrition. 131, 10-2789 S2793 S do. 10.1093/JN/131.10.2789 S. PMID 115841008. Archived from the original, abstract page, on 7 February 2009. Retrieved 2 June 2008. 6 Wang, Qingfeng, Suzuki, Azekar, Marikanda, Susanna, Paul Wallach, Stefan, Harshay, Rossika M., 1 June 2005. Sensing Wetness, a New Role for the Bacterial Flagellum. The Embo Journal. 24/11/2034. 2042 do. 10.1038/sj.embodg.76006668. PMC 11426004. PMID 15889148. 7 Albus, Sonia Vienna, Jarl, Ken F. 27 January 2015. The Archaeolum, How Archaeas Swim. Frontiers in Microbiology. 6, 23. Do. 10.3389/fmicp.2015.00023. PMC 4307647. PMID 256999024. 8 Quarks, TEF, Albus, SV, Pfeffer, F. 14 December 2018. Taxes in Archaea. Emerging Topics in Life Sciences. 2, 4535546 10.1042/ETLS20180089. PMC 7289035. PMID 3352581. 9 Hamo, LT, Rosenbaum, J.L., 1 December 1981. Cilia, flagella, and microtubules. The Journal of Cell Biology. 91, 3125S130S2, 10.1083/JCB.91.3.125S. PMC 2112827. PMID 6459327. 10-1, Kirsty Y. 21 November 2018. Coordination of eukaryotic cilia and flagella. Essays in Biochemistry. 62, 6829838 10.1042/EBC20180029. ISSN 0071-1365. PMC 6281475. PMID 3046407. 11 Streff, Stefan, Staudinger, Wilfried Franz, Merwan, Wolfgang, Easterhelt, Dieter, December 2008. Flagellar rotation in the archaeon Halobacterium selenorum depends on ATP. Journal of Molecular Biology. 384, 1182, 10.1016/J.JMB.2008.08.057. PMID 187865541. 12 AB Alberts, Bruce, 2015. Molecular Biology of the Cell, 6th ed. New York, NY. p. 942. ISBN 9780815344643. 13 Silverman, Michael, Simon, Melvin, May 1974. Flagellar rotation and the mechanism of bacterial motility. Nature. 249, 5452 7374. Bibcode, 1974 Nature.249, 73s. Do. 10.1038/249073. PMID 4598030. S2 SID 10370084. 14 Low, Graham, Meister, Marcus, Berg, Howard C. February 1987. Rapid rotation of flagella. Bundles in swimming bacteria. Nature. 325, 6105 637640. Bibcode, 1987 Nature.325637L. Do. 10.1038/325637. S2 SID 42422129. 15 Bug, Howard C. Anderson, Robert O. October 1973. Bacteria swim by rotating their flagella. Filaments. Nature. 245, 5425 38382. Bibcode, 1973 Nature.245380b. Do. 10.1038/2453800. PMID 4593496. S2 SID 4173914. 16 John, T.L., Bovey, E.C., October 1965. Movement and Locomotion of Microorganisms. Annual Review of Microbiology. 19, 12158 do. 10.1011.me.19.100165.0000321. PMID 5318439. 17 Harshay, R.M., 2003. Bacterial Motility on a Surface, Many Ways to a Common Goal. Annual Review of Microbiology. 57, 24973. Do. 10.1011.46/anov.micro.57.0305002.0911014. PMID 1452729. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 18 Ing, Sunday Y.M., Shaban, Bonnie, Jarl, Ken F. 2006. Archaeal flagella, bacterial flagella and Type 4 pili, a comparison of genes and post-translational modifications. Microbial Physiology. 11, 3. 5167191. Do. 10.1159/0000940553. PMID 169839194. S2 SID 30386932. 19 Metliner, al. November 2004. Bacterial and archaeal flagella as prokaryotic motility organelles. Biochemistry, Moscow. 69, 11 1203 1212 do. 10.1038/S10541-005-0065-8. PMID 1562737. S2 SID 632440. 20 Jarl, K. 2009. Archaeal flagella and pili. Pili and flagella, Current Research and Future Trends. Caister Academic Press. ISBN 978-1-904455-48-6. 21 McNabb, Robert M. October 2003. How Bacteria Assemble Flagella. Annual Review of Microbiology. 57, 177-100 do. 10.1046/anov.micro.57.0305002.0908332. PMID 1273035. 22 Diacy, Zoltan, Zavodsky, Peter, Namba, Kitichi, Vondervist, Ferenc, 18 June 2004. Stabilization of flagellar filaments by Hap2 capping. Feb's letters. 568, 13105109. Bibcode 2004 FEPSA.568105D. Do 10.1016/J.FEPSLAT.2004.05.029. PMID 151969929. S2 SID 33886010. 23 Gorkin, Vital D. U. Zirang, Bielnicki, Jacob, Huser, John, Ewing, Cheryl P. Geray, Patricia Igelman, Edward H. The 18th of April, 2008. Divergence of quaternary structures among bacterial flagella. Filaments. Science. 320, 5874 382385. Bibcode 2008 SCI 32382G. Do 10.1026/science.11555307. PMID 184200936. S2 SID 77020002. 24 Worrell, Liam J. Majewski, Dorothy D. Strinadka, Natalie C.J. The 15th of September, 2023. Structural insights into type 3 secretion systems of the bacterial flagellum and injectism. Annual Review of Microbiology. 77, 1669698 10.1146/An of Micro 032521-025503. ISSN 0066-4227. PMID 377134558. S2 SID 2619639668. 25 A.B. Singh, Prashant K. Shoma, Pankaj, Afanzar, Oshri, Goldfarb, Margot H. McClashina, Elena. Eisenbach, Michael, Sekchini, Gary, Iverson, T.M. The 17th of April, 2024. Cryo-EM structures reveal how the bacterial flagellum rotates and switches direction. Nature Microbiology. 9, 5 1271 1281. Do 10.1038/S41564-024-01674-1. ISSN 2058-5276. PMC 11087270. PMID 386323342. 26 Tatsumi, Tatsuo, McCartat, Linda, Ime, Yasuo, January 1992. Polar and lateral flagella motors of marine vibrio are driven by different ion motive forces. Nature. 355, 6356 182184. Bibcode 1992 Nature.355182. Do 10.1038/355182. PMID 1309599. S2 SID 4315167. 27 Kojima, Saiji, Blair, David F. 2004, The Bacterial Flagella Motor, Structure and Function of a Complex Molecular Machine, International Review of Cytology, 233, Elsevier, 93134. Do 10.1016/S0074-7696, 04, 33003-2, ISBN 978-0-12-364637-8, PMID 150373363, retrieved the 23rd of April 2024. 28 carat Dean, Tim, the 2nd of August 2010. Inside nature's most efficient motor, the flagella. Australian Life Scientist. 29 carat Whitfield, John, the 19th of June 2008. Bacterial engines have their own clutch. Nature News. News.2008.903 do 10.1038/news.2008.903. Retrieved the 17th of May 2017. 30 carat AB Botticeria, Suvic, Lopez, Shelby, Singh, Abhyuday, Harshay, Rossika M. 2024. Flagella. Motility is mutagenic. Proceedings of the National Academy of Sciences. 121, 41. Proceedings of the National Academy of Sciences. Do 10.1073/NAS.24125411121. ISSN 0027-8424. 31 carat Dusenberry, D.B., 2009. Chapter 13. Living at Microscale, The Unexpected Physics of Being Small. Cambridge, Harvard University Press. ISBN 978-0-674-03116-6. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 32 carat Hildebrand, Milton, November 1959. Motions of the Running Cheetah and Horse. Journal of Mammalogy. 44, 4481495 10.2307/1376265. Just a 1376265. Although according to Hunter, Luke, Hammond, Dave, 2003. Cheetah. Struick Publishers. pp. 37-38. The cheetah is fastest. Recorded speed was 110 km per hour, 68 miles per hour. 33 carat Meadows, Robin, the 10th of May 2011. How Bacteria Shift Gears. PLOS Biology. 9, 5E100161. Do 10.1371/journal.bio.10010661. PMC 3091840. PMID 21572986. 34 carat Sun, Chifang, Yuan, Chengjai, Zhu, Sinan, Lu, Jing, Zeng, Mtian, Cai, Zirang, Song, Hupen, 19. October 2023. Helicobacter pylori infection, a dynamic process from diagnosis to treatment. Frontiers in Cellular and Infection Microbiology. 13 do 10.3389/FSIM.2023.1257817. PMC 10621068. PMID 379288189. 35 carat Minamino, Taru, Imada, Katsumi, Namba, Kitachi, 2008. Mechanisms of type 3 protein export for bacterial flagella assembly. Molecular biosystems. 4, 11 1105 1115 do 10.1039/B808065H. PMID 189311786. 36 carat Asakura, Sho, Igachi, Goro, Tino, Tetsuo, October 1964. Reconstitution of bacterial flagella in vitro. Journal of Molecular Biology. 10/142 in 9 do. 10.1016/S0022-2836, 64, 80026-7. PMID 142222895. 37 AB Palin, Mark J. Matsk, Nicholas J. October 2006. From the Origin of Species to the Origin of Bacterial Flagella. Nature Reviews Microbiology. 4, 10/7/84792, 10.1038 /micro 1493. PMID 169532448. S2 SID 24057949. 38 AB Sire, M. March 2004. Evolution of Bacterial Type 3 Protein Secretion Systems. Trends in Microbiology. 12/3113115 10.1016/J.Tim.2004.01.003. PMID 1500186. 39 Goffner, Yuri, Ron, Eliara Z. Graw, Dan, July 2003. Bacterial type 3 secretion systems are ancient and evolved by multiple horizontal transfer events. Gene 312, 151163. Do 10.1016/S0378. 1119, 03, 00612-7. PMID 129093551. 40 McCann, Honor C. Gutman, David S. January 2008. Evolution of the Type 3 secretion system and its effectors in plant microbe interactions. New Phytologist. 177, 13347 do. 10.1111/J.1469. 8137.2007.02293.x. PMID 180784771. 41 Michael J. 2007. The Edge of Evolution, The Search for the Limits of Darwinism. New York, NY. Free Press. ISBN 978-0-7432-9620-5. 42 Rajagopala, C. Sundra V. Tits, John, Goll, Johannes, Parish, Jody R. Walbold, Katrin, McKevitt. Matthew T. Parkhill, Timothy, Maury, Hiratada, Finley, Russell L. Utes, Peter, January 2007. The Protein Network of Bacterial Motility. Molecular Systems Biology. 3/128 do. 10.1038/MSB4100166. PMC 1943423. PMID 1766750. 43 Tits, John, Rajagopala, C. Sundra V. Esther, Claudia, Hauser, Roman, Utes, Peter, November 2006. Novel Conserved Assembly Factor of the Bacterial Flagellum. Journal of Bacteriology. 188, 21. 7700 7706. Do 10.1028/JB.00820-06 PMC 1636259 PMID 169360039 44 Carcanut, Asher, Fan, Mindui, Low, Alvin W. Beatson, Scotta. Skembry, Marco. 10 May 2017. Novel genes associated with enhanced motility of Escherichia coli ST131. PLOS 1. 12, 5. E01769290. Bib code 2017 PLOS 012769290K. Do 10.1331/journal.pwn.0176290. PMC 5425062. PMID 2848982. 45 Palin, MJ, Goffner, U. 2007. Bacterial flagella and type 3 secretion, case studies in the Evolution of Complexity. Genome Dynamics. 3, 3047. Do 10.1159/0001076002. ISBN 978-3-8055. 8340-4. PMID 187537883. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 46 Bacterial flagella, PDF. Archived, PDF, from the original on 9 October 2022. Retrieved 29 December 2021. 47 Ruan, Wanfeng, Kato, Takeuchi, Santini, Clairlise, Mieta, Tomoko, Kawamoto, Akiyo, Chang. Weigia, Bernardak, Alan, Wu, Longfia, Namba, Kitachi, the 11th of December 2012. Architecture of a flagellar apparatus in the fast-swimming magnetotactic bacterium Mo1. Proceedings of the National Academy of Sciences. 109, 520643200648. Bib code 2012 NAS 109200643R. Do 10.1073/NAS.1215274109. PMC 3528567. PMID 231849885. 48 Tricho prefix. Retrieved 26 March 2022. 49 Eschezarita, Ma, Close, K.E. 2019. Vibrio flagella synthesis. Frontiers in Cellular and Infection. Microbiology. 9, 131. Do 10.3389/FSIM.2019.00131. PMC 6504787. PMID 3119103. 50 Lofo. Retrieved 26 March 2022. 51 Kudry Ashev, Mikhail, Sierklaf, Merek, Baumeister, Wolfgang, Simon, Marcus M. Wallach, Reinhard. Frisknecht, Friedrich, March 2009. Comparative cryo-electron tomography of pathogenic Lyme disease spirochetes. Molecular microbiology. 71, 6 1415 1434 Do 10.1111/J.1365 2958.2009.06613.x PMID 192106119 S2 SID 196508992 52 Kim, Yoon Kyung, McCarter, Linda L. July 2000. Analysis of the polar flagellar gene system of Vibrio parahemolyticus. Journal of Bacteriology. 182, 13 3693 3704. Do. 10.1011/JB.182.13.3693/3704.2000 PMC 94540 PMID 108509884 53 Atsumi, T. Maekawa, Y. Yamada, T. Kawajishi, I. Ime, Y. Homer, M. August 1996. Effective viscosity on swimming by the lateral and polar flagella of Vibrio algenolyticus. Journal of Bacteriology. 178, 16 5024 5026 to 10.1118.16.5024-5026.1996 PMC 178,290 PMID 8759871 54 McCarter, Linda L. 2004. Dual flagellar systems enable motility under different circumstances. Microbial Physiology. 7, 1-1829 do. 10.1111/000077866 PMID 151704000 S2 SID 21963003 55 Marino, Susanna, Shaw, Jonathan G. Toma S. 1 M. October 2006. Bacterial lateral flagella, an inducible flagellar system. FEMS Microbiology Letters. 263, 2127135 10.1111/J.1574 6968.2006.00403.x PMID 1697836 56 Bailaz, R. Simon, M. Silverman, M. July 1986. Regulation of lateral flagellar gene transcription in Vibrio parahemolyticus. Journal of Bacteriology. 167, 1-2-10-218 10.1128/JB.167.1.210 218.1986 PMC 212863 PMID 3013835 57 Canals, Rocho, Alteriba, Maria, Vilches, Sylvia, Horsberg, Gavin, Shaw, Jonathan G. Thomas, 1. M. Marino, Susanna, February 2006. Analysis of the lateral flagellar gene system of Aeromonas. Hydrophyla R3. Journal of Bacteriology. 188, 3852862 10.1128/JB.188.3.852-862.2006 PMC 1347325. PMID 164283888. 58 carat canals, Rocho, Ramirez, Sylvia, Vilches, Sylvia, Horsberg, Gavin, Shaw, Jonathan G. Thomas, Juan M. Marino, Susanna, the 15th of January 2006. Polar flagellum biogenesis in Aeromonas hydrophylla. Journal of Bacteriology. 188, 2542555 10.18028.2.542-555.2006. PMC 1347287. PMID 1638545. 59 carat Kim, Munju, Burt, James C. Van Paris, Anne-Marie J. Brewer, Kenneth S. Powers, Thomas R. 23. December 2003. A macroscopic scale model of bacterial flagellar bundling. Proceedings of the National Academy of Sciences. 100, 26 15481154855 R14, CandMat /0312562. Bib code 2003 NAS 100154881K. Do 10.1073 NAS.26335961000. PMC 307593. PMID 146711319. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 60 carat McNabb, RM, January 1977. Bacterial flagella rotating in bundles, a study in helical geometry. Proceedings of the National Academy of Sciences of the United States of America. 74, 12215. Bib code 1977 NAS 74221M. Do 10.1073 NAS.74.1.221. PMC 393230. PMID 264676. 61 carat Taylor, FJR Max, the 1st of November 2003. The Collapse of the Two Kingdom System, the Rise of Protistology and the Founding of the International Society for Evolutionary Protistology, ISEP. International Journal of Systematic and Evolutionary Microbiology. 53, 6 1707 1714. Do 10.1099 /IJS.0.02587-0. PMID 1465797. 62 carat Hulsman, Norbert, August 1992. Undulipodium, End of a Useless Discussion. European Journal of Protistology. 28, 325325 do 10.1016 /S0932-4739, 11, 80231-2. PMID 23195228. 63 carat ABCADL, Sina M. Simpson, Alistair G. B. Lane, Christopher E. Luke, Julius, Bass, David, Bowser. Samuel S. Brown, Matthew W. Berkey, Fabian, Dunthorne, Mika, Hampel, Vladimir, Heiss, Aaron. Hoppenrath, Mona, Lara, Enrique, Le Gall, Line, Lynn, Dennis H. McManus, Hilary, Mitchell, Edward A. D. Mosel Stanridge, Sharon E. Parfrey, Laura W. Pawlowski, Jan, Rueckert, Sonia, Shadwick. Laura, Skosh, Konrad L. Smirnov, Alecki, Spiegel, Frederick W. September 2012. The Revised Classification of Eukaryotes. Journal of Eukaryotic Microbiology. 59, 5429514. Do. 10.1500-115-7508.2012.00644.x. PMC 3483872. PMID 2302023. 64 Anderson, R. Ba, DJS. Lin, DH. Melkonian, M. Mostrup. Slay, Emma. February 1991. Terminology and nomenclature of the cytoskeletal elements associated with the flagella/ciliary. Apparatus in protists. Protoplasma. 164, 1318 10.1007/BF01320809. S2 SID 4075537 65 Leadbeater, Barry S.C. Green, John C., eds. 2000. Flagellates, Unity, Diversity and Evolution. The Systematics Association Special Volume. Volume 59. Taylor and Francis. ISBN 978-1-4822-6822-5. 66 A.B. Barsanti, Laura, Gualtieri, Paolo, 2006. Algae, Anatomy, Biochemistry, and Biotechnology. Florida, USA, CRC Press. ISBN 9780203492598. 67 A.B. Lindemann, C.B., Leipzig, K.A., 15 February 2010. Flagella and ciliary beating, the proven and the possible. Journal of Cell Science. 123, pt 451928 do. 10.1242/JCS.051326. PMID 2014500. S2 SID 1867350. 68 A.B. Lodish, Harvey, Burke, Arnold, Zipersky, S. Lawrence, Matsudera, Paul, Baltimore, David, Darnell. James, 2000. Section 19.4 Cilia and Flagella, Structure and Movement. Cilia and Flagella, Structure and Movement. ISBN 0-7167-3136-3. 69 Parza, Gregory J. October 2004. Intraflagellar transport and cilia-dependent renal disease, the ciliary hypothesis of polycystic kidney disease. Journal of the American Society of Nephrology. 15. 10.2528 2536 do. 10.1097/0.ASN.00001410555.57643.E0. PMID 1546627. 70 U. Buki, Naoji, Lender, Brian S. July 2013. Evolution of microtubule organizing centers across the tree of eukaryotes. The Plant Journal. 75, 223244 do. 10.1111/TPJ.12145. PMID 233982114. 71 Carat Raven, J.A. 2000. The Flagellate Condition. Leadbeater and Green 2000, pp. 2748. CRC Press. ISBN 9781482268225. 72 Carat A.B. Webster, John, Weber, Roland, the 25th of January 2007. Spores of Fungi. 2007, 3rd ed. Cambridge, Cambridge University Press. pp. 23-24. ISBN 9781139461504. 73 Carat Lara, Daniel J. G. Parfrey, Laura Wegener, Mitchell, Edward A. D. Katz, Laura A. Lara, Enrique, 22. July 2011. The Chastity of Amoeba, Re-evaluating Evidence for Sex in Amoeboid Organisms. Proceedings of the Royal Society B, Biological Sciences. 278, 1715 2081 2090. Do. 10.1098/RSPB.2011.0289. PMC 3107637. PMID 214299931. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 74 Carat A.B. Austin, CR, 1995. Grudzinskas, Georgis Gediminas, Jovic, J.L., eds. Gametes, the Spermatozoon. Cambridge, Cambridge University Press. ISBN 9780521479967. 75 Carat South, G.R., Whittick, A., 1987. Introduction to Phacology. Oxford, Blackwell Scientific Publications. p. 65. ISBN 9781444314205. 76 Carat Dodge, J.D., 1973. The Fine Structure of Algal Cells. London, Academic Press. pp. 57-79. ISBN 9780323158237. 77 Carat Lee, Re, 2008. Phacology, 4th ed. Cambridge University Press. p. 7. ISBN 9781139469876. Lee Tubular Hairs. 78 Carat Corliss, J.O., LOM, J., 2000. An annotated glossary of protozoological terms. In Lee, J.J., Leedale. G.F., Bradbury, P. eds. An Illustrated Guide to the Protozo. Volume 2, 2nd ed. Society of Protozoologists. pp. 1346-85. ISBN 1891276239. 79 Carat ABJEUCK, Alexandra, Arndt, Hartmut, November 2013. A Short Guide to Common Heterotrophic Flagellates of Freshwater Habitats based on the Morphology of Living Organisms. Protist. 164, 6. 842860. Do. 10.1016/j.protist.2013.08.003. PMID 242399731. 80 Carat Slay, M., 1989. Protozo and Other Protists. London, Edward Arnold. pp. 98-99. ISBN 9780521428057. 81 Sparrow, F.K., 1960. Aquatic Phacomistes, 2nd ed. Ann Arbor, Michigan, University of Michigan Press. p. 15. 82 Hibberd, D.J., 1976. The Ultrastructure and Taxonomy of the Chrysophyceae and Primnesiophyceae. Haptophyceae, a survey with some new observations on the ultrastructure of the Chrysophyceae. Journal of the Linnean Society of London, Botany. 72, 255-82, 10.1111/J.1095. 8339.1976.TB01352.X. 83 Slay, Ma, 1985. Origin and Evolution of Flagellar Movement. Cell Motil. 5, 137138. Archived. From the original on 3 March 2016. Retrieved 21 February 2016. 84 Cavalier Smith, T. 1987. The Origin of Eukaryotic and Archaebacterial Cells. Annals of the New York Academy of Sciences. 503, 11754. Bibcode, 1987 NYSA.503, 17C. Do. 10.1111/J.1749. 6632.1987.TB40596.X. PMID 31133114. S2 SID 38405158. Permanent Dead Link. 85 Madigan, Michael T. 2019. Brock Biology of Microorganisms, 15th, Global ed. NY, pp. 70. 71 ISBN 9781292235103. 86 A. B. Ghosh, Abrigiotti, Albus, Sonia Vierna, 1 February 2011. Assembly and Function of the Archaeal Flagellum. Biochemical Society Transactions. 39, 164-69 do. 10.1042/BST03900064. PMID 21265748. 87 Thomas, Nick Hiller. Bardi, Sonia L. Char L. Ken F. April 2001. The Archaeal Flagellum, a different kind of prokaryotic motility structure. FEMS Microbiology Reviews. 25, 2147-174 do. 10.1111/J.1574. 6900.2001.TB00575.X. PMID 11250034. S2 SID 34411164. 88 Chmielewski, Scott, Papke, Arthane, 2015. Getting a Hold on Archaeal Type 4 Pili, an Expanding Repertoire of Cellular Appendages Implicates Complex Regulation and Diverse Functions. Frontiers in Microbiology. 6, 362. Do. 10.3389/FMICP.2015.00362. ISSN 1664-302X. PMC 4419858. PMID 259999922. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 89 De Souza Machado, J. Nuno, Volmar, Leone, Schimpf, Julia, Chaudhuri, Porchelli, Kumaria. Rashmi, Van der Does, Chris, Hugel, Thorsten, Albus, Sonja Wiener, 2021. Autophosphorylation of the kite-like protein RLH inhibits oligomerization and interaction with RLI, the motor ATPase of the arculum. Molecular Microbiology 116, 3943956 10.1111/me.14781. ISSN 0950 382X. PMID 342192889. 90 Nuno de Souza Machado, João, Albus, Sonja Wiener, Dorm, Bertram, 2022. Towards elucidating the rotary mechanism of the arculum machinery. Frontiers in Microbiology. 13. Do. 10.3300/fmicp.2022.848597. ISSN 1664-302X. PMC 8978795. PMID 3538706. 91 Jarl, Ken F., Albus, Sonja Wiener, Machado, J. Nuno de Souza, 2021. A Comprehensive History of Motility and Arculation in Archaea. FEMS Microbes. 2. Stab 002. Do. 10.1093/FMSMC/PSTAB 002. ISSN 2633-6685. PMC 10117864. PMID 37334237. 92 AB Lawncore, Joyce E. Pessia, Alan P. Nichols, Donald K. March 1999. Batrachochytrium. Dendrobatidis genit sp. November, a chytrid pathogenic to amphibians. Mycologia. 91, 22192227. Do. 10.2307/3761366. JSTA 3761366. Further reading, edit. Berg, Howard C. January 2000. Motile behavior of bacteria. Physics Today. 53, 12429. Bibcode, 2000 PHT, 53A24B. Do. 10.1063/1.882934. S2 SID 1785162110. Lindemann, Charles, the 4th of April, 2008. Mechanisms of Sperm Motility. Oakland University. Archived from the original on the 16th of May, 2008. Retrieved the 18th of May, 2008. Purcell, M., 1977. Life at Low Reynolds number, PDF. American Journal of Physics. 45, 1311. Bibcode, 1977 AMJ 45, 3p. Do. 10.1119/1.10903. HDL, 2433/226838. Archived from the original PDF, on the 5th of June, 2011. Retrieved the 19th of October, 2009. Matsk, NJ, the 10th of November, 2003. Evolution in Brownian space, a model for the origin of the bacterial flagellum. talkdesign.org. External links, edit. Cell Image Library, Flagella. This article incorporates text from a publication now in the public domain, Chambers, Ephraim. ed. 1728. Cyclopedia, or an Universal Dictionary of Arts and Sciences, 1st ed. James and John Knapton, et al. Site Encyclopedia, missing or empty title equals, help. Categories, cell movement organelles, protein complexes, bacteria. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.